Hey there, hold on.
Hey, get some pie.
Good. You see me just fine? Okay, I'm gonna let you be, and don't bother Mel while she's talking, okay?
Hey, we got pie rolled on.
We'll start up in a little bit.
Delicious, oh, thank you.
Thank you, Kyle. Wherever you are. Thanks.
Do you have enough battery power on this thing?
Hey, savory pie's over there too. So there's. Yeah, that's right.
It was a lot. It's like, it was fun.
Good. Good. Melanie's defense, of course, there's pie.
Yeah. Everyone's so quiet.
Okay, thanks.
It sounds really cool. I wish that we could do this outside.
So you don't have to listen to birds in the background.
No, but the birds actually, I kind of like the
still hear
birds.
her.
He's got a mute on. Tell him to mute himself.
Hey, hold on. Can you mute your thing? Thank you. Sort of. Hey, Mark.
I'll read them all.
How are you doing? Pretty good.
Yeah.
See.
Good morning, everybody. Glad you're here. Appreciate that. Most of us know of Melanie. Most of us know her well enough to realize that she, like James, that she's round two of some of the most interesting people that have ever rolled in and out of my life here. It's hard to say everything a person can say about Melanie. I'll just say a few things before we get her started. That's the Melanie that a lot of you know. Melanie has certain sets of gifts you're all really familiar with. She really does. She cares a lot about people, and that's probably her greatest gift. Part of really caring about people is it makes her really good at meeting people. I have never seen anybody able to meet so many people at a conference like Melanie Hart. The first conference I ever took my lab guys to, this is last year, I said, hey James, how many people have you met? Nobody really. I'm like, yeah, but Melanie's met like everybody. And he's like, I wonder how she does it. And I said, she does it like this. She stands by her poster, and when people walk by, she's like, I hope you're having a good day. And she grabs them, and she makes them start talking, and she will approach anybody. And that's that line of hers, I hope you're having a good day. And I don't know if she gets that from her family, but one of the things I want to do is welcome the members of her family who are with us here today. Sam and Esther Partner in the back, and let's give them a nice southeastern welcome. Melanie was born, raised in Arkansas. She attended Arkansas State from the years 2003 to 2009. She was pacing herself. Does that sound about right? So she finished up in 2009. She's worked in a lot of different fields. She's a member of 3 different scientific societies. She's active in those societies. She, at this stage of her career, has given 8 presentations at regional and national meetings. After she finishes here, she's going to take some time and then hopes to continue her career in conservation biology. Conservation biology sort of runs through everything that she's done in terms of her work opportunities. But before we get to that, I want to show a few more images of Melanie. There's the Melanie everybody knows. There's that look. There's that frog. I love frogs, but I have this great big needle that I'm jabbing that frog in the heart with. And jabbing frogs in the heart with a big needle and drawing out what appears to be comparatively about a gallon of blood is what forms the basis of Melanie's thesis. Now, the idea of taking subject animals and sort of exposing them to certain things they're not typically exposed to apparently is not a new thing for Melanie. This is from one of her past jobs where she worked on bobwhite quail. And I only assume that's just a pose, but not a real image of what was about to happen. Graduate school for Melanie has been, many times I'm sure she's felt like this. Melanie enjoys outdoor activities, and then many times I'm sure she felt like she was barely hanging on while she was here and she was about to fall in the water, as many of the graduate students can attest to. There are times when it can be a stressful experience, and Melanie has experienced that just like everybody else. Matter of fact, I'll bet you if you were to ask Melanie 6 days ago, will you be ready for today? She would have said, I don't think so. However, I think now I think she feels differently now. She's completely ready to go. But this is an excellent picture of a quality of Melanie. Mom and Dad, is that an okay picture? Melanie is stubborn, man. She is stubborn. And today is a culmination of that stubborn quality of hers, because many times she would talk about these potential thesis projects. The first thesis project she ever talked about is the one she's going to talk about today. There are many points where I encouraged her to think about other angles, to try other things. And she goes, yeah, I just want to try this one more time. I want to work on the assays one more time. There's always saying, I will try one more time. Some of the stress that Melanie experienced while here was because she wanted to do something one more time. And when you push back those assays and try them one more time, that creates time consequences, which induces that feeling that she had in the previous slide. Now, that stubbornness means she's been able to successfully complete something that was a very hard project, and I'm proud of her for that. But my guess is she will always retain that stubborn quality. In this image, I don't know if I'm the guy on the left or the right. And if I'm the guy on the left, then I don't know who the guy is on the right, but maybe it's Roll on. So thank you for your time and helping her out, Roll on. But really what Melanie is about is hanging out with people and enjoying interacting with friends and family and church, and she's very active in all those things. She loves to be outdoors. Anytime she gets an opportunity to go anywhere or experience a new aspect of biology, she does that. For example, in terms of the field biology job she's had, she has worked for Arkansas State University, the University of Tennessee, the Arkansas Natural Heritage Program, and the Arkansas Nature Conservancy. She's worked on developing plant databases, the life history and ecology of bobwhite quail, biological control of mosquitoes During that job, she dissected mosquito larvae. So if you want to try that sometime, it sounds challenging, and I'm sure it is. She's worked on projects associated with that are about bird mortality associated with radio towers. That's part of that conservation biology theme that seems to go through a lot of Melanie's questions. She's worked on roosting ecology in bats, and she's capable of sexing bats. I don't know who else can do that. I'm guessing you do it the same way you sex a lot of other mammals, but I'm not sure.
Mm-hmm. I think so.
All right. She's worked on bird disease surveys. She just likes field ecology. Now, she likes experiencing the world, too. So here's Melanie. While she's down here, of course, she becomes familiar and entrenched in the culture of South Louisiana, and she's been very good about that. She was a person who always said, yeah, Melanie won't really get to know Mardi Gras, and she won't get to know South Louisiana, but she even proved me wrong with that. And this is just a tiny sample, but a better example of it is this. And I don't know if Mom and Dad are surprised at that or not. I was a little surprised, but like I said, Melanie will try just about anything you tell her she should try. So this is also the Melanie we know. She's completely serious. She works hard at her biology. But once she's said and done, and we're all glad she's been a part of our lives, most of us won't really remember this part of Melanie. Remember this, the crazy Melanie. That's what we'll remember. This was a day a couple weeks ago where I'm showing one of my undergraduate researchers how we collect larval Eurycia, larval salamanders up at Bogue Chitto State Park. Melanie says, can I come? And I say, dude, aren't you writing? She goes, yeah, but I'll take a little bit of time out. And I said, fine. So before we returned, we collected all the larvae, took measurements on them. We're going to release them. And Melanie says, oh, I want to take a picture. I'm like, okay, Melanie. And every picture of Melanie with an animal is like this. She has that look. So I'm eager to have Melanie show you all the things she's discovered during these 2 years and working hard on this project. And I will turn it over to Melanie, and let's congratulate her for at least getting to this point.
Thank you, Beachy, for that grand introduction. So yeah, like he said, I'm interested in conservation and in biology, and so I probably should use this. Oh yeah, the lights. Thank you. And so yeah, I wanted to talk to you guys a little bit about what I found out about traffic noise and how it can affect organisms. Specifically with the Cubs gray tree frog, because that's my study species. And a little bit about me first. I've always loved frogs. Frogs have always been my favorite animal. As you can see here, this is just like a small sample of all like the frog stuff that I've accumulated over the years. I've loved frogs for as long as I can remember. And also, I've always been interested in science, and biology has always been fascinating to me. Even when I was in elementary school, I always science was always my favorite subject in school. And so, yeah, I've always had an interest in herpetology, and actually, recently, since becoming an undergrad, I, fell in love with salamanders again. And so salamanders are pretty up there with frogs. Now, frogs do trump salamanders in that they call, and salamanders don't. But they're still pretty up there, like with frogs, as my favorites. And so I wanted to go to graduate school to pursue a career in biology and in conservation. So I wound up coming to Louisiana, which, as all of you guys know, Louisiana is a great place for looking for herps and finding herps. And I'm working with Dr. Beachy, and actually, many of you guys know just a little bit about Beachy. Many of you guys already know that he has a huge reputation of having the biggest biceps in this university. And so, and actually, I'm getting ahead of myself. I've been teaching labs all throughout my time here because I have a teaching assistantship. And so, in teaching, students will give SOTs, which is an evaluation of the instructor, and so that's helped me a lot in improving my teaching abilities and stuff like that. So it's all good, but it also completely shattered Beachy's reputation. Because proven by my SOTs from my students, they talk about my huge biceps and they tell me to get more swole. And they also tell me that I have the better biceps than my advisor. Not only that, but this student says she has bigger arms than Beachy. So the proof is here. I really like this one. Bicep size distracts from learning. Yeah. So the proof is there. But the funny thing is, for a long time he was living in denial of this. So like when it came down to it, like we were like, okay, we're gonna prove this once and for all. We're gonna have an arm wrestling match to see who has the bigger biceps. And I guess, like, I don't really understand, or at the time I didn't really know my own strength. I'm really sorry that, he wound up with a broken arm. This is him teaching herpetology, and, sorry about that. And you can tell by his expression that, he's like, oh man, she beat me again, So that was one of the reasons why I came to shed light on this subject. But in addition to that, I also, like he was saying, I have a great interest in conservation, and just, I love wildlife and I love nature, and so, I want to conserve it. And there's a wide variety of topics that you can choose from in studying conservation. And when I came here, I didn't know exactly what I wanted to do project-wise, but I knew that I wanted it to be somehow connected to conservation. And so there's like air pollution. There's invasive species like with this kudzu. That's a problem here, as well as like it's also a problem in Arkansas. And then there's like habitat fragmentation and even other things that can be caused by roads. And that brings us to road ecology, because road ecology is a somewhat new area of research and conservation. And it deals with a wide variety of topics, anywhere from like road mortality, like what's going to happen to this turtle, and light pollution. There's several lights on every road, and lights from cars too. But I was particularly interested in, I got interested in traffic noise, because with traffic noise, it's easy to work with calling frogs and work on something in conservation. Traffic noise is very widespread. As you guys know, roads and interstates are everywhere. All of you guys know that, and so it's a very large part of conservation, a very serious topic, and a lot of habitat types are close to roads, and wetlands are close to roads too. Like you see here, this is one of my noisy sites. That's very close to this road here. You can see that truck. That's on the service road. That's right next to the interstate. So this is a very noisy area where the frogs are calling. And something that's widespread throughout many taxa that use acoustic communication. Not just anurans, but also birds or any other animals using acoustic communication will change their calling characteristics in the presence of noise and in response to noise. And different species do different things to adjust their calling characteristics in the face of noise. Some species may change the frequency of their calls, call it a higher pitch than the underlying traffic noise. They may change the amplitude of the calls to get louder. They could change the number of pulses that they have in a call, change the duration or the call rate of calls. So there's a wide variety of ways that they make adjustments. And just to give you a bit of an idea, this has been firmly established in the literature that species will do this. There's even more papers than what I have listed here. So just to give you an idea, this has been firmly established, and so it's not really the question that I'm I was interested in answering by my project, I was more interested in the possible negative effects of noise. Like that, for instance, does it cause an increase in metabolic cost of calling? And if so, then, you know, would this decrease body mass more than what normally happens during the breeding season? It could also steal energy otherwise used for other purposes. And another thing is it could be stressful for the organism, either through just directly the noise effect causing stress, or it could be because of the changes in colon characteristics that they have to make in the presence of noise. And so that brings us to corticosterone. That's the hormone that indicates stress in an organism. And many of you guys heard Martha's talk, so this is kind of a review for you guys about corticosterone. And the whole purpose of corticosterone is to maintain homeostasis in the body. And it does this through a process called gluconeogenesis, which is where you draw glucose from non-carbohydrate sources. And there can be potential harmful effects of corticosterone if it's at an elevated concentration for a prolonged period of time. It can be harmful to the organism. And so how corticosterone works, it of course promotes gluconeogenesis, and this occurs in the liver cells. And so if you have a muscle cell or fat cell here, under normal circumstances, glucose will be drawn up into the cell for protein synthesis and fat production. But in the presence of corticosterone, this is inhibited, and actually the muscle and fat cells will be broken down into the amino acids and fatty acids, which go into this process of gluconeogenesis in the liver, and then the liver secretes glucose into the bloodstream. And this is how it helps animals to cope with stress. It gives the animal the energy needed to cope with the stress. But one of the adverse effects of corticosterone that can occur if you have an elevated level for a prolonged period of time is connected to this, because muscle atrophy can occur. Like it can decrease muscle mass if it's for too long that you have elevated levels of corticosterone. And so this is actually an alternative method of assessing stress, and it involves the leukocytes, which is the white blood cells, and you're counting all 5 of these cells on your blood smear. You count 100 leukocytes in all. So you're counting basophils, and this is the eosinophil, this is a neutrophil, and a lymphocyte and monocyte. And even though you're counting all of those cells, the ones directly connected to this ratio is the lymphocytes and the neutrophils. So if neutrophils increase and lymphocytes decrease, then this ratio becomes elevated, indicating stress in the organism. And it's been shown that these cells will respond in this way to a significant increase in corticosterone. And that's why it's used as a method of assessing stress. It's not well known yet, like why they respond in this way. It's not like the lymphocytes aren't being destroyed, and that's not why they decrease in the bloodstream. It's because they're being sequestered into like the lymph nodes and the and surrounding tissues in response to stress. And I mean, all of these cells respond or as a part of immunity to fight infections. So it's not clear why like stress has this effect, but it's been shown like through different papers in the literature that this will occur in response to stress. So my hypothesis is that in traffic noise sites that there will be a greater concentration of corticosterone than in pristine sites, and then in turn then there'll be a greater ratio between these cells relative to pristine sites. That the ratio will be higher. And I was using Dryophytes grossoceles, it's a Cope's gray tree frog, which this is a frog that's native to Louisiana. Normally, they'll call from different surfaces. They'll call from the sides of trees. They'll call from branches or twigs, or even from grass or vegetation. And sometimes they'll call from the ground, but they prefer to be above ground when they're calling. And if they're on top of trees, then they'll come down lower to go through in plexus, and then they'll go to the water to lay eggs. And they'll be aggressive. Males will be aggressive to each other if males get too close while they're calling. They'll call aggressively to each other. And they form large groups when they're calling in a chorus. Also, they're partial to temporary pools. You won't find them around large ponds. They're specific to temporary pools when they're breeding. And so I selected 3 noise pollution sites and 3 pristine sites. Pristine sites meaning like that there's little to no traffic noise there. And as you can see here, there's a billboard sign here, which shows you that this is very close to the interstate in the noise pollution sites. And so just to give you an idea, both of these pictures are of the same site. This is the site that you saw before with the truck in the background, and this is that service road that truck is on, with the interstate right here. And I also had to go into the woods off to the side here for some of my samples, pretty close to the road where there was a lot of traffic noise at this site. And so for my site selections, here's noise pollution site number 1, 2, and 3 here, and then we have pristine sites number 1, and 2, and 3 here. And so with my site selection, of course I had to make sure that the frogs were calling in all of those sites, and also obviously the noise pollution sites had to be close to busy roads, so all of these are close to the interstates. Also, safety played a role in my site selection. I had to make sure that, okay, are these easily accessible? Are they relatively safe to go out to late at night to do my sampling? So all of that played a role in where I chose my sites. And over here is Tangipahoa Parish, and over here, this is the only site that's actually in St. Tammany Parish. Okay, so I also went out, before I went to do my sampling, I went out with a noise level meter to assess, okay, are the noisy sites really noisier than pristine sites? And you can see that they are. They are significantly noisier. And so when I was collecting the blood, I drew the blood from the frogs through cardiac puncture, and I measured corticosteroid in the blood, and looked at the ratio of the leukocytes. And as you can see here, this frog is fine. Like, he's had his alarm ready to go back to calling. Okay, so I also made blood smears. This is me putting the blood on the blood smear slide and making the smear. And then I measured the frogs with calipers and released them to go back to calling. And in the corticosteroid and ELISA assays, this is me just doing extracting the hormone from the plasma and then using the hormone in the assay to measure the hormone. And here, this is a neutrophil, and this is a lymphocyte. And of course, I was counting the cells and figuring out what the ratios were between these cells. And so, getting into the data, with corticosteroid at least, there is not really a significant difference that I found between pristine sites and noisy sites. That was surprising to me because of course, I expected it to be significantly higher in noisy sites. And you might say that, this p-value is pretty close to 0.05. So could there be some kind of significance? But I mean, if it is significant, which it's not, but if it was significant, then it would be in the opposite direction than what was expected, because pristine sites here is a little bit higher than noisy sites. Also, I did an analysis across all of the sites, and it wasn't significant here. But I also wanted to dig down deeper because, as you see here, pristine site number 1 over here looks like it should be significantly different from at least 1 or 2 sites, at least N3 over here, which is the lowest site. So I wanted to do site-to-site F-test comparisons to really dig down deeper into this. And so I did that, and I did 15 comparisons in all. And at first glance, it looks like pristine site versus noisy site number 1, and P1 versus N3. It looks like both of those values are significant, but in order to avoid making a type 1 error, I reduced alpha by dividing 0.05 by 15, since I did 15 comparisons. And so I reduced alpha to 0.0033, and based on this p-value, there's no significant differences found. And so time to draw, I also ran an analysis here because, as some of you know, you have to make sure that you draw the blood in, you can't take too long to draw the blood from the frogs, or handling stress will become an issue with your corticosterone measurements. But here, and even though you see significance here, it's kind of strange that it's actually a negative slope. Because if there was significance, you'd expect a positive slope. But this definitely indicates that handling stress wasn't an issue for these samples. Also, I did this analysis too, which also indicates that handling stress wasn't an issue because there's not a significant difference between less than or equal to 3 minutes and greater than 3 minutes, those two groups. Because it's been, it's kind of a rule of thumb that 3 minutes or less is accepted as like to be on the safe side, you want to draw the blood in 3 minutes or less. But here there wasn't an issue here for these samples. I also ran an analysis on time of night because according to the energetic hormone vocalization model proposed by Emerson of 2001, corticosterone should increase throughout the night of nightly chorusing. But we don't see that for this species. There's not really an increase in corticosterone throughout the night. Also, frog size and frog weight. There was no relationship found here with either one of these with corticosterone. That was a little surprising because you might expect bigger animals to be able to secrete more, maybe, but there was no relationship with this species with size or weight. Also, temperature and humidity, there was no real relationship between these variables and corticosterone. But I found a very interesting interaction here between habitat type and site type. All of these by themselves, running an analysis on site type by itself is non-significant. But looking at this graph, it makes sense because, this is noisy sites and pristine sites is the hollow dots. The mean between these two is somewhere over here in the middle, and the mean between these two is also in the middle. And for habitat type, the mean between these two is somewhere in the middle, and then between these two is also in the middle. So neither of those by themselves would give you a significant effect, but the interaction between the two, when I did a two-way ANOVA, that came back significant, which is really interesting because it looks like in pristine sites, the frogs are less stressed calling from on top of grass or vegetation versus when they're on branches, whereas for the noisy sites, they're more stressed in grass and less stressed in the branches. So that's an interesting thing that I found here. It's I don't really understand the mechanism behind this on why this is happening the way it is, but it's something interesting for further research, for sure. Okay, so part 2 is the neutrophil lymphocyte ratios. One question that I had was, okay, does it have a positive relationship with core concentration? And we see that it does not actually, which is kind of surprising for me because I felt like it should have a positive relationship because, you know, as I explained before, the neutrophil lymphocyte ratios increase in response to the increase in corticosterone. But the more I thought about it, the more, you know, maybe it makes sense because something that I haven't told you guys yet about these ratios, the ratios respond a lot slower than corticosterone will respond within a few minutes, whereas, especially for ectotherms, the ratios increase, it is a lot slower, taking up to 24 hours or more for some species to reflect a significant difference in these ratios. So, given that information, these samples here that are high in corticosterone but still have a low neutrophil lymphocyte ratio, it's possible that the reason why we're seeing this with these samples is that corticosterone increased the night that I did sampling, but they didn't have a chance to reflect stress according to the ratio in response to that stress that night. And also, with these samples, having low cort and a high neutrophil lymphocyte ratio, you know, that could be because these ratios take a long time. After they become elevated, it takes a long time for them to come back down to normal, unstressed levels. It could take a few days for them to come back down. And so that could be the reason why we have a high ratio after corticosterone already came back down after stress occurred. It's not really known yet, like for any like specific species, how long it takes. But it does for the ratios to come back down, but it does take like a few days for it to come down. So the other question is, do the ratios reflect more stress in the noisy sites versus pristine sites? That was the other question, and we see that it does not. It's not a significant difference in the ratios. Now the Wilks Lambda test, I did a MANOVA on all cell types. And so the Wilks Lambda test is significant, so that, you know, that actually is because there's significance for some of the other cell counts. There's a significant difference in eosinophils and monocytes, with both of those cells being higher in noisy sites relative to pristine sites. And, you know, I didn't expect to find this. And like eosinophils mainly, their role is to fight against parasites. They also play a role in the inflammation response. And monocytes are phagocytic, and they engulf bacteria and other foreign matter. So it's possible that maybe the noisy sites are exposed to more infections than the pristine sites. I can't really, like, you know, say this for sure, because I didn't really ever measure in the frogs, like I didn't, you know, measure how, like, do they have parasites, do they not, are they infected by something? I didn't measure that, so I can't really say that, oh, there, you know, there's more infections, but it is interesting for further research, seeing these elevated levels of these cells. And I also, just like with the cort, I did an analysis across sites. This time the Wilks Lambda test came back significant, and the ratios also came back significant. So what did I do? I actually did the site-to-site comparisons again to really elucidate this. I mean, based on this graph, it looks like noisy site number 1 should be significantly different than maybe at least a couple of these sites. So I dug down deeper, and I did 15 comparisons just like before with the neutrophil lymphocyte ratios. And even though we have like, you know, some that look like they should be significant, I also set alpha to equal 0.0033 just like before, and so there's no real significant differences between any of these sites. But we did see significance for monocytes and eosinophils. Here we have N3 being significantly different than P3 and P1. And then here on this side, the eosinophils are significantly different in N3 versus P3. And at first I thought it was kind of odd that this one wasn't significantly different from this one, because it looks like it's lower, but it's because of the sample size, like the degrees of freedom is different. But anyway, yeah, so N3, you know, again, like I can't really like say this for sure, but I mean, maybe there's more infections going on in N3 versus these, you know, these sites. More research definitely needs to be done, you know, like before we can actually say that though. And what about site type and habitat type? For the corticosteroid concentrations, we saw a significant interaction here. Well, what about for cell counts? Not really. For the neutrophil lymphocyte ratio, there was no significant difference from these. As you can see here, these are pretty close together, so there was no interaction found here. Also, I ran an analysis on size and weight in regards to the ratios, and there was no relationship found for these, just like there wasn't for quart. Same thing for temperature and humidity. There was no relationship for either one of these. And so in conclusion, both core concentration and neutrophil lymphocyte ratios agreed that traffic noise, at least for this species, it doesn't stress the male Cope-Spray tree frogs. Now, that may not be true for other species. There have been other studies conducted indicating that there was an increase in corticosteroid in frogs that have been looked at, and in other animals like birds, in response to stress or in response to traffic noise. And so maybe it's like dependent on the species that you're looking at. But we found that interesting interaction between, you know, noise, depending on habitat type, that you know, noise had an influence depending on what the frogs were calling from. But there was no relationship with quart with the neutrophil lymphocyte ratios, and there was no interaction. And just like I said before, you know, there might be higher rates of infection in some sites. You know, maybe not. More research needs to be done on that. So the one question is why? Like, why would it not be stressful for them? Well, it could just be because they're just not stressed out by noise, and for this species, noise has never been stressful for them. Another possibility is that they could be becoming acclimated to the noise, or they have become acclimated. You know, maybe if it was stressful for them before, by being out there in the sites, they've been breeding there for a really long time, and the traffic noise has been there for a long time. So they could have acclimated to the noise, and so they're not responding, you know, with quart to the noise anymore. But more studies are needed, you know, of course, to elucidate all of this, because, you know, like with this study, you can't really know which one it could be. Another call out for further research: someone should evaluate the energetic cost of calling and see whether there's a difference between noisy sites and pristine sites. That was something I was really interested in doing for my project, but because of time constraints, I really couldn't do it. But that would be really interesting to see for different species. And so with that, I wanted to thank thank you so much, like everyone who came. I really appreciate that. To hear me speak, and like I especially want to acknowledge my my committee members, Dr. Beachy and Dr. Valverde and Dr. Fontenot. They've helped me tremendously with this project. Without them, I wouldn't have been able to do any of it. So really, like I'm very thankful for all three of them. 100%. Like they're awesome. And also everyone that helped with field work. Thank you so much to those people too. That really helped me out. And especially Jeremy Rodriguez, like he came out with me a lot, like pretty much twice a week he came out with me, and that helped tremendously because I was, you know, like this was night field work, and some sites, you know, aren't safe to go out to by myself, and so I really wouldn't have been able to go out to those particular sites without the help of these people, so I really appreciate them. I also wanted to just say that without the plate reader to read the assay plates, there's no way that I could have even done the assay. So I really appreciate that from Dr. Childers and Dr. Shockett providing that. Also, I just wanted to real quick say that Whitney Walkowski has helped me a lot. Like I really appreciate her because she's like, you know, through email. Like, some of you guys don't know her. She was a previous graduate student that did work on pig frogs with corticosterone and with testosterone. And so she, like, was a great source of information for me with doing my project. And always, the biology graduate students, my experience here has been a lot fuller with all of you guys. I really appreciate you guys. And I'm going to miss everyone. Like, I've really enjoyed it here. I'm kind of sad to leave, actually, but, you know, so I appreciate, like, just the experience I've had here with all of you guys. And, of course, my family, moral support and everything. And so with that, I can take any questions. Whoops. Oh, Oliver.
Did you notice any like abrupt differences in frog population density between the pristine sites and the noisy sites?
No, I didn't really notice anything there. They seem pretty much the same, but like I didn't really measure that, so I'm not really sure. Yeah.
How much of it do you think had to do with vibrations as opposed to like the actual noise itself?
Oh.
I know that vibrations affect animals a lot.
Yeah, that, you know, I've heard a lot about that, you know, with avians too, with frogs, that, you know, vibrations do affect, like, you know, stress and stuff like that. I'm not really sure. That's a really interesting thing that you brought up. I think it would be interesting to look into that. I'm really not sure. Like, some of these sites seemed like they were far enough away from the sites that vibrations wouldn't become a factor. But, you know, I don't know, like, you know, how far vibrations would go. So that would be interesting to look into.
When you do your lymphocyte ratio, are you just picking out a square area on your slide and then just counting within there to do it? Or?
That's a good question. Like you have to, like, you have your blood smear and you have to go to the feathered edge, which is like where the red blood cells are evenly distributed, like kind of in that area, and it's easier to count cells and more accurate in the feathered edge. And so you go in a zigzag pattern in that area, counting all of the leukocytes that you see in each field of vision, like as you're going, until you get to 100 cells, and then you stop, and that's like you take measurements from there.
Are these animals aphagic during their mating period?
What do you mean by aphagic?
Do they stop eating during that period while they're mating?
I don't think so. I don't think that they do, because this species like breeds for a prolonged period of time. Like they're breeding all summer long. Mm-hmm. So even like during the breeding season, they're self-eating. I'm pretty sure.
By default, would you not expect the pristine sites to have greater level of biodiversity more competitors, more predators than your roadside sites, your I don't want to call them degraded sites, your less pristine sites? Did you see differences in just general diversity across those two areas?
Not really. I didn't really see a difference. I know that like with one of my pristine sites, I remember like I saw a snake out at pristine site number 1 is out at North Oak Park, and so you know out there I saw snakes and stuff. But in all of the sites, I guess you know I was kind of assuming that's one of the assumptions I had to make, that they were pretty similar across sites as far as predators are concerned. But I didn't notice any differences.
The reason I ask is one explanation, because your corticosteroid results are opposite of what you expected, right? That perhaps competitors or predators, more of them are higher stressor in your pristine site than what you would see in your road site. Especially the road noise may have an impact, but the fact that there's more competitors calling or other predators there more on the pristine site may overrule the road noise.
Yeah, that's a possibility. That would be interesting to look into.
Has anyone ever shown that these animals are capable of discriminating that difference in sound intensity that your machine recorded? Do they know that it's louder next to the road compared to the other place?
Well, you know, I don't know of any like specific papers on that, but I know that like you know, like frogs do have the ability to like you know, with their tympanum, like they're able to like you know tune out certain frequencies and stuff like that. So like it could be.
I mean, measuring intensities, you didn't look at the frequencies at all.
Yeah, I like the. I think that they would be able to tell, you know, like that, and like I didn't really measure their ability, but they, I think that they'd be able to tell between like whether there's a greater level of noise in one side versus the other. But that's something that I should read into more. Other questions?
Did you measure the noise every night you went out, or did you just measure it like before you started your field work?
I measured it before I started my field work.
Because I was gonna say it would have been interesting if you could have done like a relationship between like noise that particular like per night versus corticosteroid, to see if there's like any sort of relationship between.
Yeah, that would have been a good way to do it. Like part of the reason why like I didn't do that was because of like time constraints, just because like you know it would have taken a long time to take like the measurements from like everywhere in the site and then go out to take the samples, but that would have been, that's a good idea to do that in the future. Penny.
So in any of the examples where noise has been shown to be a stressor and caused like lymphocyte ratio changes, corticosterone increases, have they shown any effect on infection in the stressed animals, like vortex?
That's a good question. That I don't think that has really been looked into. So that's actually a good area for further research. Like I think that should be done. But no, none of the papers that I've looked at really looked at that. They just looked at the stress.
Okay, thank you, everyone. Make sure you grab some pie on the way out.
Yeah, yeah. Oh. Oh, thank you.
Yeah. Well, dude, I just read a paper that like
Thank you. I really enjoyed it.
Oh, sorry. That's how they
No, thank you. And I'm trying to live stream it. I don't know how well it worked, but hopefully maybe like people can watch. Go to YouTube and watch it. I think Ross was saying that.
Yeah. Tell her.
Oh,
Tell
yeah,
her. She's the one that gave you that pie. You better tell her thank you.
Yeah. Thank you.
That's water.
Thank you.
Okay, so you didn't
Oh,
mention.
I've been watching this. You
Just go over that same story. Yeah, I was asking, like, how do you?
It's super easy.
I think we're doing like,
And you're not even.
I mean, it's not.
Oh.
To do like multiple time samples.
Well, that's not gonna happen.
So that way you could like compare if it was like due to like noise pollution or just due to.
Oh, you are so well.
I'll come out. Like sampling.
Oh, no, can't say anything.
Or you talk about like that.
You're like, so take like your initial sample and like second one or something just to
Yeah,
see if there's
because you're thinking that maybe there would be like if the level was already elevated, then it wouldn't increase as much.
It would, but
like the second time.
Yeah, no, but like.
You guys have any left?
Because maybe, so like your stress or something happens due to something else other than like
So see you later.
Thank you.
Good job.
Bye, Melanie. Thank you.
Melanie, good
You're
job.
welcome.
Good job, kid.
Well, these two were store bought because I didn't really have time to make them, but the quiches are homemade and these are homemade.
Did you think there might be any relationship towards reproduction in animals?
Like
With
between,
corticosterone?
like that corticosterone was inhibiting.
Not necessarily inhibiting, but actually
It's,
I know in certain eel species, corticosterone facilitates it because they go eat catches. They're not here where they're reproducing.
Yeah. Well, you know, what's interesting about like what I've learned about like frogs, especially males, is that as they're calling, and there have been a few different studies that have shown that corticosterone has this relationship with calling, where as the frog is calling and using the energy reserves,
off the ground, but not off of the
eventually,
below.
like corticosterone
Pretty
will
high,
increase and
if we
reach a threshold level where it inhibits calling, and then as corticosterone decreases and the energy reserves come back up, it'll resume calling like after enough corticosterone has been metabolized. And so, you know, frogs will sometimes go through like calling and satellite behavior. Go back and forth.
So you don't think there's any true relationship with the physiological effects of reproduction?
Only with, I think with a lot of frogs it actually inhibits the reproductive actions. You know, like at least for the males. You know, for the females it might be different. Yeah, thank you. I'm gonna go.
So, and then I was thinking about how many people are looking for the lessons. Like, why don't we look at the 20 frogs at 30 miles? Agency would say now it's been downgraded to 20.
But we're getting the eastern side.
Water, right? So we'll get some more.
Well, last week we went on.
There were a few frogs that weren't calling, but for the most part,
No, I mean, if I did, I've got different exams until 1 o'clock.
yeah.
So, dark,
Everyone.
and then try to come back in the dark.
Right.
Or salamanders or whatever.
Yeah, sure.
Came again here.
This will, this next part of
Once I get out.
Oh, yeah, that's true. And also with Coach.
Thanks for coming, Rob.
Very good, very good. Nice
Yeah.
job, Melanie.
Thank you.
Is he gonna make
Glad I
you
was
work?
able to hop in.
I'm gonna go to the bathroom.
Okay.
Today.
And once you come back, Mel, we'll get started.
They'll come.
Oh,
Oh, I probably should.
My son Ryan here is hanging out with me. But, yes, we certainly enjoyed hanging
Hi, well done.
around. So.
Well, she loves.
Took my Boon and safety class.
Okay, we'll get started in a few minutes here,
I
okay?
never got to use any.
Mel has to
coach
use
yet.
the restroom and we'll be ready soon.
She got offered